Welcome to the APFCB webinar on the role of clinical laboratories in COVID-19. In this lecture, Dr. Donal, Huda, Nasil will be talking on the molecular point-of-care testing for COVID-19.
Good day, everyone. I'm Huda from National Public Health Laboratory Malaysia. Thank you APFCB for inviting me for this workshop, and my topic today is molecular POCT for COVID-19. And this is outline of my presentation, a bit on the timeline about COVID-19 and on the molecular POCT role, what assay we have for detecting the virus, and some of the lists that are available, what we do in Malaysia, and the future recommendation, as well as the challenges and way forward. Okay, this slide showing the timeline of COVID-19, or initially we call it nCoV. On the 31st of December 2019, WHO China Country Office was informed that there was a cluster of pneumonia of unknown origin, initially occur in this seafood market. About 41 persons infected with this pneumonia, and on the 1st of January, this market was closed down. Okay, and as you can see from this timeline, the first case on the December 8, the first 41 cases. And then on the 10th of January, the whole genome sequencing was shared by the scientists from Fudan University. And after the sharing of the sequence, all the scientists all over the world actually started to design primers for detection of this novel coronavirus. Okay, let us look at this novel coronavirus. It's actually a group of beta coronavirus. I'll show you later what are the other groups. Okay, and it causes us fever, dry cough, dyspnea, and bilateral ground glass opacity. On the chest CT scan, the clinical actually resembles SARS and MERS. Okay, and it has incubation time that varies from as early as 1 day, but can prolong up to 14 days. And asymptomatic patient also can spread the disease. Okay, what happened in my country, Malaysia? Okay, for the first 3 cases that we identified on the 23rd of January 2020, actually those are the contacts of the tourists that has been confirmed in Singapore on 20th of January 2020. And the Ministry of Health from Singapore contact us to actually do the contact tracing, and 8 of the contacts was taken the samples and sent to the lab that I work, Mama Kesihatan Awam Kebangsaan, or National Public Health Laboratory. And this is when the first 3 cases identified in Malaysia. Okay, but this slide showing the current situation. This is up as per August 30th, 2020. All over the world, it's already identified or detected more than 25 million cases Okay, and then 846,000, more than 846,000 cases of death already occurred up to August 30th. And as you can see from the slide, US being the case with the highest number, the country with the highest cases number, with the highest death as well. Death is already 186,857. And this is in Malaysia. Up to 31st of August is already 9,340 cases with 127 deaths. Okay, and then the chart below showing the cases throughout the month of August, which as you can see, the cases are below 50 cases. The average is around 10 cases per day. Okay, all right. And for this slide, I will recall us a bit on the respiratory viruses. This slide showing all the common respiratory viruses that we usually encountered, such as influenza, parainfluenza, okay, RSV in children, metapneumovirus, quite rare, adenovirus, and as well as coronavirus. Okay, and about this coronavirus, is actually an envelope RNA virus distributed broadly among human, mammals, birds, and it causes respiratory, enteric, hepatic, and as well as neurological disease. Before this novel coronavirus, actually there are 6 coronavirus species that are known to cause human disease, such as 229E, OC43, and L63, and HKU1. These are the coronavirus that cause common cold, even in immunocompetent individuals. And for SARS-CoV and MERS-CoV, those 2 viruses cause more severe illness. It causes severe acute respiratory syndrome, and it also linked to sometimes fatal illness. Okay, SARS-CoV in 2002 to 2003, and MERS from 2012 and until now. Okay, where is the coronavirus? Okay, this slide actually shows the virology of coronavirus. It is included under RNA viruses, like I mentioned, positive stranded, envelope, and other envelope viruses such as flavivirus and toga virus. Okay, and as you can see from the below diagram is a diagram of a virus which has proteins, and as you can see, the spike proteins is protruding from the virus, okay, as well as the other virus that are the used for the detection of virus later. Okay, and from the left picture, it shows the size of this coronavirus, and the comparison between the other viruses and also some of the bacteria. Okay, the classification of the coronavirus, okay, it's from the family of Coronaviridae, okay, and the genus, as you can see, it can be alpha, beta, gamma, and delta, but SARS coronavirus is under beta coronavirus. Okay, about the transmission, droplets and contacts actually are the two method of transmission. Okay, from the droplet, it can caused by causing, sneezing. Okay, from the contact such as touching a person or touching an object contaminated with the virus, and later if you touch your mouth, nose, that is when the virus can enter to your body. Okay, and rarely it can also be from fecal contamination. Okay, and clinically, coronavirus can cause from mild, moderate, and as well as severe illness such as pneumonia and bronchitis. Okay, what are the diagnoses that can be done to diagnose COVID-19? Okay, the method of diagnosis, first is real-time RT-PCR. This is actually the gold standard. Okay, but you can also do sequencing, but of course it is time consuming and more expensive to be done. Okay, and then you can also do serology tests, but of course serology tests, for example, antibody detection can only give you whether the patient has been exposed to the virus, but cannot diagnose whether you are currently having the virus. The antigen serology test actually can give you the whether the patient having the illness or the current infection, but currently there are still not many tests from the antigen detection that is sensitive enough. Okay, so this one showing the gold standard method for diagnosing COVID-19. Okay, this is taken from the interim guideline. Okay, real-time RT-PCR, which can detect the presence of RNA, the viral RNA. It has high sensitivity and specificity, but of course the sensitivity can decrease over time. That is when your testing cannot detect the virus if the patient come during the late onset. And of course the quality of the sample as well can decrease the sensitivity. Okay, as I mentioned just now, on the 10th and 11th of January, the scientists from the China already shared the whole genome sequencing, and WHO also has recommended these two protocols to be used for detecting the virus. One is from Berlin, and then the other one is from Hong Kong. Okay, and this one example from Common et al. Sharing the protocol for detecting COVID-19, which it detects the gene of RDRP gene, E gene, as well as N gene. And this is how those three genes are located at. Okay, the RDRP from the ORF1AB genes. And the other method that you can do to detect initial COVID-19, actually virus isolation. If you don't really know what virus are you dealing with, you can do viral isolation first, and then after that you can do confirmatory via RT-PCR or sequencing. Okay, and this slide is showing on the left side is the Viro E6 cell lines, and on the right side is the infected cells, which showing the rounded, or we call it cytopathic effect. Okay, and then electron microscope also can be done from this picture. Actually, this is taken from the first 3 cases that detected in our lab. Okay, for this electron microscope showing this coronavirus with size of 76 nanometer. Okay, and sequencing can be used for diagnosis as well as for the epidemiology purpose. And as you can see, this sequencing also been shared to the gene bank, like in this one, GSAID. Okay, and then the scientist also has compared the Wuhan SARS-CoV virus with the other virus, such as the virus from the bat precursor, from the pangolin precursor, as well as SARS-CoV 2003, which only has about 80% similarity. And this also can be done to look for the epidemiology link of the virus that has been identified all over the world. Of course, there will be a little mutations, and still there is correlation with the first case from the Wuhan, China. Okay, this is, as I mentioned, serology test, which, for example, the antigen and IgM and IgG that only can be detected up to the first day 5 to day 14 of infection. Okay, now we proceed to the molecular POCT. Okay, rapid and accurate testing significantly contribute towards epidemiologic surveillance and future outbreaks such as infectious disease. And in-house testing of RT-PCR is endorsed by WHO as well as from the CDC, and adopted by many reference labs. But during outbreak, when healthcare resources are insufficient to combat the situation due to increasing number of patients and demand, point-of-care testing shall be regarded as essential for testing viral genome nucleic acid, and this can reduce the chances of spreading of the infection and also prevent from the delay of reporting. Okay, what is POCT Molecular? POCT Molecular actually is a portable molecular test which is a rapid, accurate, and cost-effective detection device. The roles and the advantages: it can give you rapid and timely diagnosis. It does not require a trained or expert professional such as in conventional RT-PCR. It is low in the cost. It can be robust and economical for diagnosis for COVID-19. It can also give you early detection, and of course, the early detection can decrease the case and also control the outbreak, and also it reduces transportation needs, and thus it can lower the spread of infection. Okay, what are the molecular assay that can be used to detect viral nucleic acid? Okay, from this slide, I'll show all the molecular assay that are available for detection of viral nucleic acid. But the red font are those available in the form of point of care testing, such as RT-PCR POCT, the reverse transcriptase loop-mediated isotomal amplification, CRISPR-based assay, and microarray. Okay, let us go to each of the testing method. Okay, first RT-PCR. RT-PCR actually a method of amplifying a tiny amount of viral genetic material in a sample, and it is for this COVID-19 a gold standard for diagnosis. Okay, samples can be from respiratory specimens, from serum, stool, ocular secretions, and current paper has shown that saliva also can be used to detect COVID-19. Okay, and this slide showing the method of RT-PCR. First, you take a swab from the oropharyngeal or nasopharyngeal swab, and then the samples are sent to the lab. Okay, and then in the lab you do extraction by purifying the RNA, and then the process of converting the RNA into copy DNA by reverse transcriptase, and then master mix process, which you put all the primers and probes, okay, as well as the enzymes, okay, and then you put all this mixture in the PCR machine, which is a thermocycler, and then the PCR machine will duplicate the virus DNA, and the dyes bind to the copied virus DNA for detection. Usually the cycle could be up to 40 cycles, and for detection you can see from the graph, it's either when it positive it will give you that sigmoid curve, but below picture is the negative. Okay, what are the limitations of RT-PCR? So why can't we do RT-PCR in all cases? Okay, RT-PCR actually require expensive lab instruments. Okay, for example, you need a special lab to do the RT-PCR. Okay, and it also need a skill. You need to train the personnels. Okay, and then results can also took quite some times. Okay, as early as 6 hours, and it can also be up to days if you have problem in the QC and in the technical issue, everything. Okay, and do we have RT-PCR for the point of care testing? Of course, we have, for example, Gene Expert and CASTAT, which use RT-PCR to diagnose COVID-19. Okay, for expert, from the Gene Expert Express SARS-CoV, it's a molecular in vitro diagnostic test which detect SARS-CoV-2, okay, and use nucleic acid amplification technology as RT-PCR, but it contains all the primer, probes, and internal controls, and it can detect qualitatively from the specimen. Okay, the detectant genes are E and also N2. This is how the cartridge of Gene Expert looks like. Okay, the result can be obtained as soon as 30 minutes. Okay, but now there's also 4-in-1 test expert that detects SARS-CoV-2, flu A, flu B, and RSV for let's say the patient have dual infection. And the other testing machine is from the company of Sensure. Okay, this is another molecular POCT called iPonetic. Okay, it detects the ORF1AB gene and N gene. Okay, and this only need one instrument for all the extraction, master mix, and amplification techniques. Okay, and it took about less than 40 minutes with the machine weight less than 10 kilos. So it's easy to carry, and it can be put at the remote area or the small clinics. Okay, the next method is isothermal nucleic acid amplification. Okay, as you know, the RT-PCR requires multiple temperature change for each cycle. But for isothermal nucleic acid amplification, as the name is only done in constant temperature, so it doesn't need a thermocycler. Okay, one example is reverse transcriptase loop mediated isothermal amplification, or RT-LAM. Okay, it is a rapid and cost-effective method. It requires a set of 4 primers specific for the target gene or region to enhance the sensitivity, and it will be combined with LAM with reverse transcriptase tab to allow for detection of RNA. Okay, and detection is done via photometry. This is only require heating and visual inspection. Okay, so it is sensitive method, and it also is a promising candidate for virus detection. And this is how the methods of the loop mediated isothermal amplification, or LAM. Okay, and the example of the diagnostic method using this technology is ID Now from Abbott Diagnostic. Okay, it says that it only took less than 13 minutes. Okay, sample is upper respiratory specimens, and per test only can do 1 sample. Okay, this is how the machine looks like. Okay, and it shows the benefit of easy to use, minimal training, okay, and good performance, and reliable and near point of care testing. The other method, but this actually is not point of care, but a method to isothermal amplification technique to detect the virus as well. Transcription mediated amplification, it amplifies a specific region, either RNA or DNA. It is as much as efficient of RT-PCR, which use retroviral reverse transcriptase and T7 RNA polymerase. Example of this platform is Hologic Pentafusion platform. Okay, it has the capacity to perform both RT-PCR and TMA simultaneously with a throughput up to 1,000 tests in 24 hours. Okay, and it can also screen the other common respiratory viruses using the same patient sample and collection vial. And the other method is CRISPR-based assay. Okay, what is CRISPR? CRISPR is clustered, regularly interspaced, short palindromic repeats. Okay, it's actually a family of nucleic acid sequences found in prokaryotic organisms such as bacteria. Okay, and these sequences can be recognized and cut by a set of bacterial enzymes called CRISPR-associated enzymes, such as Cas9, Cas12, and Cas13. Okay, and certain enzymes in Cas12 and Cas13 families can be programmed to target and cut viral RNA sequences. So from this method, it can be used to detect the viral RNA. And these two companies, Mammoth Bioscience and Sherlock Bioscience, established the technique of CRISPR method. Okay, the Sherlock method use Cas13, okay, and then the detector use Cas12. And this is how the method of the CRISPR protein looks like. Okay, the advantages of CRISPR method, it does not require complex instrumentation. It can be read using paper strip, okay, and can detect the presence of SARS-CoV without loss of sensitivity and specificity. The cost is low, and it can be performed in only within 1 hour. So this is one of the great potential method for point of care testing for COVID-19. Okay, for this Sherlock CRISPR, actually it already received the FDA authorized for EUA diagnostic test. Okay. Alright, the other method for isothermal is rolling cycle amplification. This is also not actually a point of care, but it can be performed under isothermal condition with minimal reagent and avoid the generation of false positive result frequently encountered in PCR-based assay. Okay, for microarray, actually it's a rapid high throughput test. It rely on the generation of copy DNA from viral RNA using reverse transcriptase, and subsequently can label the cDNA with specific probe, such as RT-PCR. And the label of cDNA are loaded into the well of microarray trays containing solid phase oligonucleotides fixed into the surface. And if they hybridize, they will remain bound after washing. And after washing the unbound DNA, the signal can presence and the signal showing presence of the virus-specific nucleic acid. The advantages of nucleic acid hybridization using the microarray, it can also identify mutation up to 24 single nucleotide polymorphism. Okay, and also it is one test to rapid detect the strain or the mutation variation. And this is how the mechanism of microarray, as I mentioned just now. Okay, an example of the platform using this method is from BioFire COVID-19 test kit. Okay, this is also this picture is showing us on the March 23rd. It still has not cleared FDA, but I think up to now is already have approved by FDA. Okay, so it's easy to use. Okay, as you can see, only one machine. Okay, fully automated, and minimal equipment and consumables needed. Time taken only 45 minutes. Okay, and I'll show you also the other method of detecting by sequencing. This one, amplicon-based metagenomic sequencing. It's actually a dual approach using amplicon-based sequencing and metagenomic sequencing. Okay, metagenomic sequencing is used primarily to address the background microbiome of infected individuals. Okay, it rapidly identify SARS-CoV virus and also other pathogens that contribute to secondary infection. And it allows for potential contact tracing, molecular epidemiology, and studies of viral evolution. And these are the three methods which can be used from the sequencing. First, amplicon and metagenomic mean iron based sequencing. Second, next generation shotgun metagenomic sequencing platform. And the third one is NGS with molecular barcode. Okay, what are the current available molecular assay for detecting SARS-CoV? The current method are mostly by RT-PCR, which 90% of the technologies use this method. 6% use isothermal amplification technology. 2% use hybridization technique. And another 2% use CRISPR-based technology. And some of these tests are actually high throughput platform or have a short time to result and therefore are in high demand. Okay, and this slide showing the list of the POC molecular that are available worldwide. Okay, for example, ID Now that I mentioned. Okay, BioFire. Okay, Expert, and some of the method using the CRISPR technique. Okay, and then CASTAT actually is a rapid multiplex RT-PCR, but only took about 1 hour, which use E and RDRP genes. And the other method you can find from the Find website. Okay, those are the list of the POCT or near POC NAT to diagnose COVID-19. Okay, in Malaysia, actually, we do have requests for point of care testing being done. Okay, but all the device that can be approved being used in Malaysia need to be request for the emergency use under the special access of COVID-19 testing at the Medical Device Authority, Ministry of Health. And in MDA, they can do evaluation and correlation tests with the gold standard testing, and it is done in the Ministry of Health evaluation lab, such as in my lab as well. Okay, and then all the recommended device or test kit are later be listed to the Malaysian Medical Association or MME, and the detection genes of the kit is as recommended by WHO. Okay, what are the future recommendations for COVID-19 diagnosis? Actually, for future, we need highly reliable, accurate diagnosis, point of care with high sensitivity and specificity, high positive predictive and negative predictive value to combat different infectious disease in the future, and also in this current pandemic COVID-19. It is all still now needed in both the developed country and as well as the underdeveloped country over the world. Okay, what are the challenges and way forward? Challenges now is to scale up the reliable testing, okay, to increase the capacity, to design an affordable, easy to test method, okay, reduce dependent on the lab molecular facility, okay, and then accessible testing to the field use, to the remote area as well as to refugee camp, okay, and testing also need to be as accurate as RT-PCR with high sensitivity. And this also can reduce the lab burden and meet the goals to screen, detect, and isolate the patient to stop this pandemic. I think this is the last slide. With that, I thank you for your attention.